very good. Okay, first of all, a couple of announcements. I mentioned in class yesterday that I was going to go over the phylogenetic groupings assignment with you all online today, but I actually think I'm going to wait and do that tomorrow in class. I think I'll probably get more out of it if we're all together in the same room. So I'll delay that till then. What I'm going to do today is finish up chapter 25 and then actually move on to chapter 27, which is going to be on the next exam. It's the prokaryotes. So your exam that's coming up, what is it, Monday, I believe, right? It must be Monday. Yeah, next Monday. That's right. So your exam next Monday is going to be through the chapters 22 through 26. So what I'll be covering in the later part of the lecture today will be chapter 27. That'll be on exam 2, but we'll go ahead and get a start on that. But let me finish up chapter 25, and then we'll go ahead and jump into that. So where we had finished off last time in chapter 25 was right at that moment when the Cambrian explosion occurred. And this is really significant into the diversity of life on the planet, because prior to this point, as I mentioned in my last lecture, there were animals around. You can see in this particular picture, sponges, which are animals, have been around since, looking at the scale, over 635 million years ago. Sponges have been around for a while. The jellyfish, that's the cnidarians, the C is silent in that word. They had been around since about 600 million years or so. But what we mean by the Cambrian explosion is not the first appearance of animals, but actually this explosion in diversity. There was obviously no real explosion. So it was this explosion in diversity. You can see in the chart the Cambrian explosion, which, you know, it varies in dates. I got up here 535 to 525 million years ago. My pen decides not to work as soon as class starts. Thank you. So I got this up here. Remember, this is one of the things that bothered Darwin, because Darwin only understood evolution in the sense of gradualism, which does work. But we talked about punctuated equilibrium too, where you can actually get a rapid diversification of species, especially if it involves a group of islands, like we talked about with Darwin's finches. But the problem that Darwin had, of course, was the fact that in the fossil record at about this time, it seems like these animal groups, to him anyway, appeared all at once. Now, obviously, that's not the case. The Cambrian explosion didn't happen in one day. As you notice down below, it was a rather long fuse. It happened anywhere between 25 to 100 million years. Now, does that mean that some people say it lasted only 25 and some say it lasted only 100? No, what we're talking about there is it lasted anywhere between that amount of time. And some folks even go a little bit longer. Again, it didn't happen overnight. So somewhere around, I usually kind of remember the number 520. It always helps me. About 520 million years ago, we went from a few animal groups, again, the sponges and the cnidarians for the most part, to pretty much all the modern animal groups that we have today, the stuff that we'll talk about later on in the chapter. So the echinoderms, the sea stars, the chordates, and that's where we belong. Some smaller groups like the brachiopods, the segmented worms, the arthropods, and even those groups of animals that may have had members around before the Cambrian explosion exploded in diversity after the Cambrian explosion. And there's always been a lot of speculation as to what caused this explosion. You remember, like I said, with Darwin, it was problematic for him because it seemed to happen very quickly, and he wasn't aware of that punctuated equilibrium aspect, the fact that devolution can occur relatively fast sometimes, especially if the environment is changing relatively quickly. But what was driving all this, for the most part, was, we think, this idea of predation, what we call an arms race. And if you think about it, as organisms develop the ability to feed upon one another, like jaws begin to develop and teeth begin to develop and organisms got bigger and all that sort of stuff, that would drive a pretty quick arms race. In other words, as predators become better at consuming prey or attacking and killing prey, if the prey, of course, don't want to go extinct, then they're going to have to find better ways to combat the predators. Either they're going to be faster, or maybe they have protective coatings, or are able to hide better. And then, of course, this is what we mean by the arms race. As the prey got better at hiding from the predators, well, then the predators had to get better at finding the prey. So it becomes this back and forth. That's what we mean by an arms race. The predators evolved to become better predators. The prey evolved to become better prey, or not being preyed upon. Of course, now once again, let me remind you that we only talk about the success stories here. That a lot of organisms during this time, during the arms race, most likely went extinct. In other words, predators got really good and they didn't evolve quick enough to survive. And of course, when you don't survive, then extinction is what's in the future for you. All right, so we think it was pretty much this predator-prey relationship. And even in today's world, when we look at organisms and evolution and organisms out there in the world, we find that predator-prey relationships are one of the biggest deciding factors in driving diversity, right? Trying to stay alive so that you can reproduce and produce offspring, all that stuff we've been talking about all along, is an extremely important part of perpetuating the species on the planet. All right, so you can see why that would be a very strong environmental pressure. And of course, once again, this is the environment. It may not be the height of the leaves on the trees that the giraffes and Lamarck was talking about. It may not be anything like the weather, but anything in your environment, including something that's trying to eat you, is going to drive selective pressure, right? So that's what we mean by this whole idea of an arms race. And we think that's what occurred at about this time during the Cambrian explosion. So it really drove this idea of diversification very quickly. Now, one of the other things that happened during this time, so we're talking about the animals here, let's not forget about the plants. One of the other things that happened during this time, about 500 million years ago, so right during or right towards the end of the Cambrian explosion, was that plants began to move out on the land. All right, now, prior to this point, there wasn't much or anything on land by which organisms that weren't photosynthetic or organisms that weren't autotrophs to feed upon, right? And of course, if you're not a photosynthesizer, you've got to have something that is in your environment in order to live. So during this time, about 500 million years ago, plants began to move out on land, began to colonize land, and that really opened up opportunities for lots of other things. Not only the organisms that ate the plants, right? Now the heterotrophs, critters like you and I, could move out on the land and maybe even move a little further away from the water because they had things to eat out there. But plants also have a way of changing the environment, of changing the soil, if you will. And in many cases, what plants will do is they'll actually change the soil chemically in such a way that makes it easier or more conducive for other species to colonize land. Now, of course, plants don't do this because they're trying to help each other out. You understand that plants don't have that ability to do that. But again, it's this happenstance of chemistry that occurs. So as these plants were moving out on the land and doing their plant thing and modifying the soil, those modifications allowed opportunities for other species of plants to get a foothold. So, for example, there are certain groups of plants out there that we call the legumes. Legumes are the beans, the peas, peanuts. Peanuts are a legume. Peanuts are actually not a nut. They're actually a legume. Legumes plants are very good at putting nitrogen into the soil. And nitrogen is an extremely important element for plant growth. We often say that nitrogen is the limiting factor in plant growth. When you hear discussion about a limiting factor to an organism, what we mean by that is usually the resource, it could be nitrogen, whatever the case may be, that is in the shortest supply, that is in the least supply. So that's the thing that really decides whether organisms like plants can survive or not, the amount of nitrogen. If you fertilize your lawn or your flower beds or any plants that you might fertilize, fertilizer has nitrogen in it, and that's an essential ingredient for plant growth. So legumes, as they may have moved out on the land, what they would have done is they actually deposit nitrogen into the soil through a symbiotic relationship. And now that extra nitrogen in the soil would have created other opportunities for other plants that don't have the ability like legumes do to survive, right? So it's kind of a, like I said, it's a happenstance. No plants are doing this for the benefit of other plants. But because of this particular plant, we get this particular result. And because of this particular result, we get other plants that can move in. Now, another thing that helped plants move on the land, and we'll talk about this when we get into the fungus chapter, is this stuff called mycorrhizae. Kind of a weird spelled word. Mycorrhizae is a fungus, all right? And oftentimes we think about a fungus, especially in terms of plant growth, as being a bad thing. Fungi can be a bad thing. Fungal infections can kill organisms, including plants. But there are not only just bad fungus. Obviously, there's not. You eat fungus, right? Mushrooms aren't bad fungus. Mushrooms that you eat. Some mushrooms can be bad fungus in terms of what they do to you. But this particular fungus actually grew around the roots of plants. Let me show you what I mean. If we take a picture, a microscope picture of a plant root, this part is the root. All right, so just to give you an idea, if this is the root of the plant, what they've done in this picture is they've cut a section through it. All right, so they sliced a thin section, placed it down on a microscope, that's the root of the plant. And all of this stuff that you see on the outside that's surrounding the root, that's mycorrhizae. That's a fungus. So what we mean is the fungus and fungi, which will, again, like I said, when we get into the chapter, fungi grow with little threads all over the place. They're not really roots because only plants have roots. But all over this plant root, there's going to be these little threads. And we'll find out when we get to the fungus chapter that fungi have a very interesting way of extracting nutrients from their environment. But anyway, that way that they do that actually is beneficial to the plants. Now, once again, fungi and the plants, there's no, mental, there's no mental interaction between them. But the fungi derive a benefit from growing on the roots of the plants. Okay, and then the plants gain a benefit by the fungi growing on their roots, by the mycorrhizae growing on the roots. So again, it's a symbiotic relationship. You help me out, and I'll help you out. Now, more about the mycorrhizae when we get into the fungus, but in terms of what it did for the diversity of life, especially on land, is these fungi, these mycorrhizae, in conjunction with the plants, allowed plants to more easily colonize land. All right, they made it easier for plants to extract nutrients from this new soil, this new dirt, right? This stuff that had formed, this land that had formed. Another thing when plants moved on the land, another not side effect, but another beneficiary of this was the fact that the arthropods increased in diversity rapidly, about 450 million years ago. Now, we, of course, will talk about arthropods later on, but arthropods are insects, okay? Insects, crustaceans, shrimp, lobsters, bumblebees, arachnids, spiders, ticks, mites, scorpions. These are all arthropods. But specifically the type of arthropods that really benefit from plants moving on the land were those arthropods that were in a relationship with plants, the pollinators, okay? We all know, of course, that bees move from flower to flower, and the bees do that because they receive nectar from the flower. And what the flower receives is the fact that the bee, as it moves from flower to flower, transmits pollen. Pollen is the male part of the flower, so that's where the sperm is. So when that bee flies from flower A to flower B, it's moving the sperm from one flower to the other flower and potentially fertilizing that flower. So the relationship between especially flowering plants and insects is a very close one, the pollinating insects. Not all insects are pollinators. So when the plants moved out on the land, and then insects found this opportunity to live on land, and in conjunction with these pollinating plants, everybody benefited. Right, the insects gain a benefit because the plants provide them something to eat, the nectar, and the flowers gain a benefit because they have something to move their pollen around. Pollen can be transmitted by wind in a variety of different areas, but insects is one of the ways it's done, and it can be done pretty effectively that way. So we kind of, remember, this is all hand in hand. So plants are moving out on the land, and they were able to do that because these mycorrhizae are really important, folks. Most all plants today, if you examine them, still have these mycorrhizal relationships. They still have these things growing around the roots. They changed the soil, the plants did, for other plants, like I was talking about the legumes. And then, like I said, the arthropods, right? The arthropods being especially the pollinating insects, not to mention, of course, the fact that now the arthropods moving on the land had something to eat as well, right? Many insects, of course, eat plants. Now, the biggest problem with moving out of water onto land should be obvious, right? It's desiccation. You're going to dry out. In the water, you don't have to worry about drying out. But when you move out on the land, that becomes a situation. one of the things that we see in these early plants that evolved were these waterproof coatings on the leaves of plants. There are even plants around today that still have that. Mangrove plants that, you know, the ones with the big prop roots. Let's see if I can even, so here's a mangrove, sorry, right? And they have these big prop roots that extend. If you've ever been along, which you have, I'm sure, along the coast or down in the Keys, you know what I'm talking about. The water is up in there, right? The leaves, especially of the red mangroves, are thick. They have sort of a waxy coating to them. And the reason why that is mangroves can grow in water that is salt water or brackish water. In biological terms, what we mean by brackish is it's not salty like salt water, like the ocean, but it's not fresh water. The ocean is about 3.5% salt. Brackish could be anywhere between, you know, 0, really, that would be fresh water, and 1 or 2 or 2.5% salty. So the salt concentration can vary. But salt dries things out. So growing in salt water would desiccate you. What red mangrove's done to compensate for that is they have this protective coating over their leaves, kind of like when you put chapstick on, right? It keeps the moisture in. Now, this desiccation problem or this desiccation issue is something that not only plants had to deal with, but also animals. So when we start to get into animals moving into land, we start talking about the different adaptation of land, of animals to land, you'll see this word desiccation again. That's a real important difference, right? That's a real important thing right there. If you move on the land, you're not living in water, your biggest problem is going to be drying out. Not only you drying out as an adult, right? But also your offspring, and that's going to drive a big change in eggs, which we'll get to when we get into the animals, all right? So here we are at about 500 million years ago. Plants are moving to land. The arthropods are moving to land. Everything seems to be working well. Diversity is increasing. Organisms are spreading out to different environments, right? Remember, the advantage of spreading out to new environments is you got the place to yourself. So you have all the resources in that new environment, however limited they might be. If you can extract those resources and you're one of the limited few, then it's all yours, right? And that would, of course, be beneficial to these organisms. But as life goes on Earth, it's not always hunky-dory. It's not always a good day on Earth. Sometimes we have extinctions. We recognize in the geological record 5 major mass extinctions. You can see them on the chart here. Here's one. There's a second one here. This is a big one. We'll talk about this one. It's 3, 4 right there. And then this one we'll also talk about. This is when the dinosaurs went extinct. So this is the Permian extinction up here. That was a massive extinction. Look at that. About a third of the planet's species were wiped out. And then this is what we call the Cretaceous extinction. And that's when the dinosaurs. Right, 2 of the major extinctions. Recently, there's been talk of a 6th mass extinction. And actually, that would be the extinction that's being caused by habitat destruction by human beings. So we may be in the midst of a mass extinction event, all driven by human beings destroying the environment, right? But we'll, like I said, we'll focus our attention on the 2 big ones, the Permian and the Cretaceous. Right? So let's look at the Permian extinction first. So we're at about 240 to 250 million years ago. So this is about 250 million years after the Cambrian explosion. So here's the Cambrian explosion. Everything looks good. There are some extinction events you can see, but we're moving our way up here to about 250 million years ago to the Permian extinction. All right, during the Permian extinction, like I said, it was a very large event that occurred, a very large extinction event. About 90% of all marine species went extinct. About a third of all insect orders went extinct, and it happened pretty quickly in about 5 million years. Extinction events very often form the boundaries between these eras, like the Paleozoic and the Mesozoic. All right, so here we are, and geologists very often separate the eras based on a variety of things, and one of those is extinctions. So that break between the Paleozoic, and always remember the Mesozoic is the dinosaur era, all right? So what we're talking about here is the extinction event that occurred right before the dinosaurs began to rise to prominence. Now, what caused this extinction event? We know what caused the dinosaurs. That was a big asteroid. But what was it during the Permian extinction? Well, one of the things that happened about this time was the formation of a huge landmass known as Pangaea. So let me show you what Pangaea looks like. So at about this time, here we are, about this time, 240 to 250 million years ago, this is the way the continents on Earth looked. And we say that during Pangaea, during the Permian extinction, there was the formation of Pangaea. So land began to develop, right? Land masses began to form. Volcanic eruptions that began to deposit material. That material began to accumulate on the ocean floor, and as it got more and more, it eventually broke the surface of the ocean, and we formed dry land. All right, so during the Permian time, this was happening. Well, you're thinking, well, why would that drive extinction? Because that actually seems like a good thing. We're making more land, more diversity of habitats, and we just talked about diversity of habitats would be beneficial for species diversity, right? Different habitats, different species can exploit that. Well, you know, just because it's good for one set of species doesn't mean it's good for another set of species. So during this time when Pangaea was forming, there was a major disturbance to marine and terrestrial habitats. Specifically, and I've listed them on the screen here, one of the things that happened during this formation of Pangaea was the oceans got deeper, and the inshore water areas were reduced. All right, what we mean by that is prior to the formation of Pangaea, as I was talking about the volcanoes producing land, there were little chunks of land. And after that, as these little chunks of land got bigger and bigger, they formed this huge continent of Pangaea. Now, even today, most species diversity in the marine environment, most ocean species, most of that diversity occurs around land. There is not a lot of species diversity out in the middle of the ocean. And one of the reasons why that is there's not a lot to eat out in the middle of the ocean. Because we go back to this idea of what plants do for an ecosystem. All right? Plants are the food producers. Every ecosystem has to have the producers. Every ecosystem has to have something that takes the energy from the sun, just like you learned in Bio 1, and turns it into food. If you're not autotrophic, if you're a heterotroph, then you're limited. So most of this productivity that was occurring was around near inshore waters. And again, it's that way today. One of the reasons, of course, is the fact that inshore waters are shallower. And so producers that need sunlight could grow in shallower water. There's no light at the bottom of the ocean, right? Light attenuates or fades out in the ocean pretty doggone quickly. So the deep ocean is not going to have a lot of productivity. I mean, there is some. There's the photosynthetic plankton. But a lot of the productivity is going to be around land masses. And then running off the land would be a lot of essential nutrients that plants need to survive. So like I said, even today, most of the productivity that we have on Earth is going to be mostly around the land masses, with a lot less of it being out in the middle of the ocean. Well, when these continents continued to grow and finally grew together, that diminished the amount of coastal area. That diminished the amount of inshore waters. So for those organisms that evolved to survive in these inshore waters, you can see the difference between these little blobs of land that I drew over here on the left, and now we got this big continent of Pangaea. There was a lot less shoreline. So those organisms would have been impacted. They would have lost their shoreline. The formation of Pangaea would have also changed a lot of the way the currents in the oceans would have flowed. And organisms, of course, that's a change to their environment. That could have affected them as well, negatively and positively, depending on the species. And then, and this is kind of the beginning of the dinosaurs, or how the dinosaurs made a living. Dinosaurs developed a way, reptiles did, to actually remove their dependence on water, especially for reproduction. Without getting too far ahead of myself, we all know that amphibians and frogs and toads, they go back to the water and lay their eggs, right? They lay their eggs in the water and they become tadpoles, which have a tail. They lose that tail, they grow legs, and they move out on the land. You can't get too far away from water, though, if you're an amphibian. For one thing, your skin is not really well developed. I mean, some are. The toads are a little bit better, but your skin is not really well developed for that desiccation problem. And more importantly, your eggs have to be laid in water. Reptiles, for reasons and for physiological reasons that we will talk about when we get into them, developed the ability to lay an egg that did not depend on water. All right, so they could lay their eggs away from water. And since they could lay their eggs away from the water, reptiles could begin to move into the interior of the continents. Now, in this picture, you can see the yellow part in the interior of the continents is reflecting the fact that, and it's just the laws of climatology and weather, interiors of continents are going to be drier. All right, and that interior continent being drier means it would drive evolution. Organisms that could live in these drier climates because of their eggs, because of their skin, right? Reptiles developed the ability to produce a skin that prevented that desiccation. You would open up more opportunities for a diversity to occur. Now, for those species that were living around the coastline, let me erase my scribbling. So let's imagine a situation where we have a chunk of land here and a chunk of land here. We got all this shoreline. These species that lived in here, when these two chunks of land grew together, well, their habitat's gone, right? So they would have went extinct. But the organisms that had the ability to move into the interior part of the land, they would have found a habitat unencumbered, in other words, by themselves again, that they could have diversified into. Again, remember, most of the species went extinct. As I said, about 90% of all marine species and about a third of all the insects went extinct. A lot of the reasons we think maybe the insects went extinct or could have went extinct could have been because we lost a lot of plants back then. Remember that relationship that occurs between the two of them. So this whole disturbance to ecosystems, both in the terrestrial and in the marine environment, could have led to one of these massive extinctions. But in biology and in the history of life on Earth, the detriment of one group of species very often drives to the rise of another group. All right, so as we move on from Pangaea, from 250 million years ago, the continents begin to drift apart until we get into the present day. So we moved to about 250 million years ago. Then we moved up into two major land masses, Laurasia and Gondwana here. And then we're continuing to drift. And of course, the continents are continuing to drift. These continents are moving that way. And the Atlantic Ocean is getting wider. Pacific Ocean is getting narrower even today. It's not moving very fast, just a few inches a day. So that's why we're looking at millions of years here. So Pangaea broke up. And of course, as it broke up, then that would change habitats as well. Another thing that was happening during about this time, 250 million years ago, there's the plates that I'm talking about. These are the crustal plates on Earth that are moving. So, you know, we're on the North American plate and it's moving off to the west. The Eurasian plate is moving off to the east. And then over in the Pacific, they're all gathering. That's why there's so many earthquakes in the Pacific. That's what these little red dots are. There's a lot of plate movement. And when plates come together and they crash into one another, they can dive below. One plate can move below another one. They can move together and actually form a mountain. Out in California, the two plates move side by side. So any of that forces like that can cause earthquakes. And a lot of that is occurring around the Pacific Ocean. We actually call it the Ring of Fire around the Pacific Ocean because of all the volcanic and plate tectonic activity that's going on. And of course, during a lot in these areas, you also have volcanoes. And we're familiar with volcanoes. We've seen volcanoes erupt, right? And when volcanoes erupt, they do an awful lot. They change a lot about the environment. Not only the noxious gases that they produce. Remember, we were talking about life began on Earth. We said the early Earth was, you know, it was kind of a rough place. There was a lot of volcanic activity. And there's a lot of nasty gases that the volcanoes put out that are detrimental to life. There's also the idea that volcanoes, you know, all the debris gets up in the atmosphere and blocks out sun, which affects the photosynthesizers, which affects the organisms that live on the photosynthesizers. So what volcanoes can do to the environment is pretty obvious, right? But what I want you to understand about this is during this time, there was a tremendous amount of volcanic activity. There wasn't just a few volcanoes every once in a while that erupt that we read about in the paper. a lot of the world was still very volcanic. You remember the early Earth was very volcanic and it began to calm down. But during this time, and throughout other times, volcanic activity would occasionally rise to prominence. So the Earth would be relatively calm for a while volcanically, and then there would be a whole bunch of volcanic activity that would erupt. And when I'm talking about volcanic activity, it's not just in volcanoes. We're talking about situations where's that picture? There we go. We're talking about situations in which you would have these massive volcanic areas. Now, I'm sorry this is in Russian. This is the best map I could find. But what's highlighted in the blue is an area that's now called the Siberian Traps, T-R-A-P-S, Siberia, because it's in Siberia. These are the Siberian Traps. And what this is highlighted in blue is this is a massive, I guess the only way to explain it really is like a lake. This is a massive lake of volcanic activity. So imagine like in the picture I have up in the upper left, you got the volcanoes, but you have these huge lakes of lava. So look how big that is too. I mean, Russia's a huge country. So we're taking up half or more of the country is just this massive amount of lava and volcanic activity. So we're talking about spewing a tremendous amount of material into the atmosphere. So the Permian extinction today, geologists believe for the most part was caused by two major factors: the formation of Pangaea and the changing of the habitats that occurred, and again this renewed volcanic activity that just made the world a much more difficult place to survive. Now, of course, as I've said, just because you make the world more difficult for some species doesn't mean you don't open up the door for others. So after the Permian extinction, after the volcanic activity began to settle down again, the environment had been changed, right? As I said, about 90% of all marine species, about a third of the insects went extinct, and that's just a couple of examples. A lot of other species suffered as well. But there was one species, or there was one group of species or group of organisms after the Permian that were able to take advantage of all this new habitat. And of course, that is these guys. There we are, the dinosaurs. Right? The dinosaurs took advantage of that change at the end of the Permian extinction, of that change in habitat. And since, of course, dinosaurs are reptiles, and as I mentioned, reptiles overall developed the ability to live in drier climates or more diverse climates, and the world had those now, we get this major rush of the dinosaurs. And I don't know how many species of dinosaurs there were. There's probably some 8-year-old out there that can tell you that, but there were a lot of different kinds of dinosaurs. There were dinosaurs in the ocean. There were dinosaurs in the air, and of course there were all the land dinosaurs. That's that part in Jurassic Park I think about that I love, the first movie where he goes out amongst when he first sees all them dinosaurs and they're running around and he's standing amongst them and he noticed that they're kind of flocking like birds. That's really kind of cool. All right, so we get the dinosaurs, and the dinosaurs rule for 200 million years or so, and of course, don't think that the dinosaurs kept the mammals from evolving. There were already mammals during this time. Mammals had evolved early in the dinosaur era, but they really didn't rise to prominence because the dinosaurs were the head animals in charge at this time. They were the dominant ones. They were big. Talk about the arms race. Look at that Tyrannosaurus in the middle, right? Lots of big teeth, the ability to consume other organisms. So the mammals during this time were relatively small, and they pretty much didn't come out during the day. They were nocturnal. It came out at night. Because it came out during the day, the dinosaurs would feed upon you. But it was the dinosaurs that were the dominant for 200 million years. And they diversified into a variety of different habitats, and they got bigger and bigger and bigger, like the Tyrannosaurus and the Brontosaurus that we're all familiar with now. You can get bigger and bigger and bigger because there's a lot of resources, a lot of things to eat. And of course, if it's an arms race, the bigger you are, the stronger you are, and heck, the more teeth you got, yeah, you're going to be one of the top predators, right? Everything's going very nicely for the dinosaurs for 200 million years. They're diversifying. They're enjoying their dominance on the planet. And if the next event that we're getting ready to go into didn't happen, it's likely the dinosaurs would still be prominent on the planet. And the mammals may not have been. In other words, the event that happened next, the one that caused the extinction of the dinosaurs, probably we owe our existence, humans, to this event. If the dinosaurs hadn't gone extinct and the mammals hadn't had the chance to have the place to themselves, then we might not have had human evolution. It just blows me away. Because whereas the Permian extinction was a combination of events that kind of accumulated through time, you know, the land moving around and changing habitats, all that volcanic activity, the extinction of the dinosaurs literally did happen in one day. The current theory about why the dinosaurs went extinct, or what we call the Cretaceous extinction, about 65 million years ago, half of the marine species, many families of terrestrial plants and animals, all the dinosaurs went extinct, happened in one day. It was an asteroid strike. You see in the picture in the upper right, there's an asteroid about the size of Manhattan, by the way, so no small rock. This was a really, really big asteroid. This asteroid, when it hit the Earth, released explosive energy well beyond any of the nuclear bombs that we've been able to develop. Well beyond. I mean, it would make a nuclear bomb look like a firecracker compared to what happened when this asteroid hit the planet. It's fascinating. You can get online and see some of the simulations of what it was, but when this big rock hit the planet, it just peeled away the crust of the Earth like an orange, spread fire and just brimstone and nasty everywhere. So this was a major event. Now, what I want to talk about in the Alvarez theory is how this was found out. When I was a kid growing up, that was the question. Where did the dinosaurs go? If you look in the fossil record, the dinosaurs literally disappeared overnight. All right? It's like, what happened to them? Well, the Alvarez theory is a couple of, I think it was a father and a son from the University of California, Berkeley. When they were looking through the rock layer, they noticed this dark band that I'm trying to show with the red arrow right there. Right there, this dark band. That dark band is called the KT boundary. The K actually means Cretaceous, but it's Cretaceous in German because it was first discovered, I believe, over in Germany. So they spell Cretaceous with a K, but that's the KT boundary. The T is the next period, the next era that occurs. So this is during the dinosaurs. So you find dinosaur fossils here, and then you find none here, separated by a very thin boundary. So when they looked at that boundary, they said, what is it? What's in that boundary that may give us a clue? And what's in that boundary is an element, a high concentration of an element known as iridium. Iridium is one of the elements on the periodic chart. You can look it up. But the thing about iridium is rare on Earth. That's a very rare element. However, very common on asteroids. All right, so that's what gave the Alvarez crew there, I think it was a father and a son. That's what gave them the idea. All of this iridium that you find in that little thin black layer. Oh, by the way, no matter where you go on Earth, no matter where you dig, at about the same time, 65 million years ago, you find this black layer of high concentrations of iridium. All of that iridium that we see in that layer throughout the Earth came from that original asteroid. So when that asteroid hit the Earth, and of course it disintegrated into a million pieces, and all those pieces rained back down onto the Earth with all kinds of devastation, as you can imagine, the iridium settled in this nice little thin layer about 65 million years ago. So that's our evidence that it was an asteroid. And this has been pretty well supported in the scientific literature. And we even have, of course, the big thing is an asteroid that big should have made one heck of a crater, right? Now, meteor craters and asteroid craters on Earth are going to be a little difficult because the Earth has erosion. And most of if you look at the moon, right, the moon is pockmarked with all kinds of meteor strikes and asteroid strikes. And there's no reason to believe that the Earth didn't suffer that many, but there's no erosion on the moon. So those evidences on Earth disappear after a while. But recently, just within the last 20 or 30 years, 40 years, we have found evidence of a crater. It's the Chicxulub crater, and it's found just off of the Yucatan Peninsula. So they've done some geological surveys, and they found that there's a crater that part of it's on land and part of it's in the water that's just about right there. Which you think if that were to hit today, that would really screw us up right here in Florida, wouldn't it? Anyway, that asteroid smacking the Earth. You can see these guys without even understanding what's going on. They're having a nice day in the water. Everything's good. Here comes that asteroid. Boom. Changes the Earth. Literally in one day, right? Changes the Earth, wipes out the dinosaurs. And the dinosaurs are gone. Now the mammals have a chance, right? Now not all the reptiles went extinct, just the dinosaurs. So there were still the reptiles, the amphibians, but, and I know this is very concentrated. This is in your book as well. So we're right there. And if you go right there and look what happens next. After the dinosaurs die off, at 50 million years ago, we get the radiation. Radiation, we're talking about species diversification among birds, mammals, pollinating insects, flowering plants, and those two things would have went together, the pollinating insects and the flowering plants. And then a little bit later on, 25 or so million years later, we start to get the first primates, and then here come human beings. So that's what I mean about that. I mean, without that Cretaceous extinction, without that asteroid, it's very possible that the world that we see today would have been a lot different than we do see it today, especially among the mammals. All right. Now, are there other asteroids out there that can cause mass extinctions on Earth? Oh, yes, absolutely. NASA tracks a lot of this stuff. Presently, according to them, there's no asteroids that have us in the crosshairs, but it's reasonable to assume that we will probably be hit by a major asteroid of that size sometime in the future. NASA actually has these interesting names for asteroids. They size them by their destructive ability. So they call them, I'm trying to remember, I think it's called city killer or town killers, city killers, planet killers, depending how big the asteroid is. It could wipe out a city or it could wipe out the entire life on Earth, as it will. All right. Recently, I heard that the biggest problem, too, or the biggest concern we have now is not so much the asteroids, it's comets. See, we can track asteroids from quite a distance away. So if we see one that's got us in the crosshairs, there are techniques, theoretical, but there are techniques perhaps to divert it off its path so it misses us. But comets, on the other hand, you can't see comets until they get within about the orbit of Jupiter. And the estimates I've heard was that if we detect a comet just about where Jupiter orbits that has us in the crosshairs, we have about 3 months until that comet hits us. That's not a very long time. So it's the comets that's the big thing too, right? Asteroids would be a problem, but we'd be able to at least track them. Now, what we mean by this radiation, as I said, is adaptive radiation, right? So what we see right after the mammals, or right after the dinosaurs died off, right about here, actually, right about there, we see this rapid diversification in mammals. Not so much in the monotremes, like the platypus, which we'll talk about, but the marsupials advanced or radiated, and then the placental mammals like you and I radiated. All right, so we get this just extinction of one species and this radiation of another species. Now, it didn't just happen in animals or mammals. It also happened in plants. And very often it happened on islands. There were a lot of these regional adaptations that occurred in an area, like we have looking on the Hawaiian Islands. We find that about 5.1 million years ago, we see evidence of this particular species that's on the Hawaiian Island today. And then these other species on the other islands are relatives of this original species. And what they've done is they've moved out onto a group of islands, and because each of those islands might have been slightly different, they diversified. So this is Darwin's finches among plants, right? So these adaptive radiations that occurred on Earth after the dinosaur extinction would have been both worldwide and also in regional areas, like we see among these guys. All right, so then we get to the modern day, and we have the diversification that we have today. Now, as we go through the different groups of organisms, plants, and animals later on in the lectures, we'll get in a little more detail about the changes that occurred in the radiations. But in a nutshell, that's kind of where we are today. Dinosaurs go extinct, plants, pollinating insects, mammals, radiated. Other organisms also evolved, those that made it through the extinction. And we end up with the diversification we have on Earth today, which to me, again, is fascinating because remember I told you during the Cretaceous, 90% of all marine species went extinct. So that's what I mean by that is what you see today on Earth, the diversity of marine organisms is the remnants of the 10% that didn't go extinct. It's amazing to me to just think about the diversity involved in that. All right. Let's change gears for a second and talk about how we could get organisms changing quickly through time. So after the dinosaurs go extinct, what are some of the other contributors to this rapid diversification and radiation that we see of species? Well, let's go back and look at developmental genes. Now, developmental genes are genes that control the development of an organism. And we're talking about development in the organism as it grows, especially developmental genes that control things that occur during the embryonic development. It's a little like saying, well, that's not a good example, but if you make little changes early on in an organism's development, that can have very major morphological, that can result in major morphological differences later on. Developmental genes control, number one, they control the rate and the timing at which things happen. So as an embryo is developing, these developmental genes control when that happens, how fast or slow that happens. And developmental genes also control spatial patterns. So it controls where different body parts occur. All right, so changes in those developmental genes could have major changes in how species evolve. And the two big developmental genes that I want to discuss are developmental genes that control things called heterochrony, which is the rate and the timing. A chronograph. A chronograph is a watch. So chronological order, right? So hetero means other, so changes in timing or rate. And then something we call homeotic genes, which I'll talk about in just a second. Homeotic genes control spatial patterns. All right. So let's take a look at heterochrony for a second. How does that affect things? All right. We know that human beings and chimpanzees are related, relatively closely related. So if we look at the skull of a chimpanzee fetus and we look at the skull of a human fetus, they're very similar to one another. And suffice it to say that if I had a skull of a human fetus and the skull of a chimpanzee, then unless you were learned in this field, you might not be able to tell the difference between those two. But of course, the skull of an adult chimpanzee and the skull of an adult human are very different. What we're talking about in terms of developmental genes, these heterochrony genes, is if these genes change the rate at which different parts of the skull grew, you would get the differences that you see in the chimpanzee and the human adult. All right, so we started here, and of course, the chimpanzee would have grown a little bit a quicker, faster rate in the jaw region, which made that more prominent jaws that we see in chimpanzees and other primates that we see in human beings. The skull in chimpanzees would have grown and become actually, would have kind of lengthened out, whereas in the human adult or in the humans, it would have become much larger for a larger brain. So you see what I'm talking about? Changes that have occurred in the fetus. You don't see anything different in the fetus, but the rate and the timing of those changes could have resulted in very big changes in the adult. Okay? So that could have been something that occurred among a bunch of species. Changes in developmental genes very early on that had very dramatic changes early on in the adult. Even if you look in that chimpanzee infant and the adult in the picture, especially in the jaw region, very different. I mean, chimpanzee infants their skull look very much, if you shaved that thing on the left, it would look kind of like a human in a way, wouldn't it? But I don't know too many humans that look like the adult on the right-hand side. Now, other things that would have occurred in heterochrony would have been this interesting thing called paedomorphosis, all right? First of all, we start with this idea of the changes in the timing of reproductive growth relative to somatic growth. All right, let's review for just a second. Somatic cells are the cells in your body or the cells in an organism, the regular cells. You have somatic cells and you have reproductive cells, right? You got sperm and egg and the cells that create those through meiosis. And then you have the other cells in your body which are created through mitosis. Now, the change in the timing of reproductive growth. In other words, what we're saying is if the reproductive cells begin to mature and grow faster relative to the growth of the other cells, it's a process that we call neoteny. What we might get is something called paedomorphosis. And paedomorphosis basically means that the adult retains ancestral juvenile characteristics. Paedomorphosis means the form of a child. Let me show you a picture of what I mean by that. Salamanders are amphibians, and salamanders develop in the water, and when they develop in the water, they have gills, right? And they have a tail that's very suited for getting around in the water. Once salamanders develop into an adult, they lose the gills and the tail slenders down and they become something that looks like this. This is the slimy salamander, Plethodon glutinosus. We have these in Florida. You can find them in the Wekiva River. Now, the one in the bottom, though, the big picture, that's an axolotl, okay? Ambystoma mexicanum is this particular one. You notice there's a couple of things about this. It's got the gills. It's got front legs. It's got smaller, more rudimentary rear legs, and it has that very distinctive tail. This is not the juvenile form of the axolotl, though. This is the adult. All right. So what we mean by this idea of paedomorphosis is an adult axolotl looks like a juvenile of other salamander species, and this is the way axolotl spends its life, even when it becomes reproductively mature. So that leads to the next part of this. If you begin to be able to reproduce in the juvenile form, now let me make sure I clarify that. You're not a juvenile, okay? Because if you're a juvenile, you're not reproducing, but you look like a juvenile. This paedogenesis refers to that ability for something that looks like a juvenile, like the axolotl there on the bottom, to reproduce. So if organisms through time developed or begin to develop this idea of paedogenesis, then we get a rapid change in the morphology of the organisms. And this again would be controlled by genes and the rate and the timing. So as organisms are developing as a juvenile, if the reproductive cells matured faster relative to the somatic cells, you would get an organism that looks like a juvenile but is able to reproduce. Now, in this particular case, we get these species of salamanders, these axolotls that are basically adults but look like other juvenile. A plethodon, a slimy salamander juvenile, would look very much like the axolotl with the gills and the rudimentary hind legs and the tail, which would then change as that organism begin to, like frogs do. They lose their legs and move out on the land. So the same thing with the axolotl. So the changing in the rate and the timing of those genes could end up producing organisms that are able to reproduce when they look like a juvenile. Now, make sure you understand they're not a juvenile because by definition, juveniles don't reproduce, but they look like a juvenile. All right? Now, other things that could have caused rapid diversification of organisms would have been the homeotic genes, the genes that control the position of things like appendages. Among homeotic genes, there's a group of homeotic genes that we call the Hox genes, very important among animals. Hox genes, as I said, control positional information. During the embryonic development of the animal, they control where the legs and the appendages are going to be found. So I have an example here for you. About 400 million years ago, a mutation to a Hox gene, specifically the UBX Hox gene. There are many different Hox genes, okay? For example, in insects and crustaceans, there's the UBX and the SCR. All right, sorry about that. Got disconnected for a second. So where was I? Okay, so we're talking about Hox genes. We're looking at an example of how we went from swimming versus feeding appendages in crustaceans. So down here below, there's a crustacean known as Artemia, and Artemia's got many different legs. All right, Drosophila, the fruit fly, has much fewer legs. So what we're saying happened here in these homeotic genes among animals is about 400 million years ago, there was a mutation that occurred to the UBX Hox gene. And what that mutation did, in Artemia, the UBX gene actually does not suppress leg formation, so you get a lot of legs. And that's what we see in crustaceans. Crustaceans have lots of legs, right? Think about lobsters. Think about shrimp. Not only the legs in the front, but think about all the legs that are in the tail of the lobster. Those are swimmerets. Those are still, maybe not call them legs, let's call them appendages. Insects have far fewer. So this mutation, what it did in the UBX gene is among insects, now the UBS gene suppresses leg formation. The end result is, or the bottom line is, we get a situation where we get a group of organisms that now have much fewer legs. And this mutation that occurred to the UBX gene is the responsible for that. And now having fewer legs, we begin to diversify those appendages. And when we talk about the arthropods later on in the semester, I'll come back to this, but the arthropods are all about the appendages. They're all about modified appendages. Antennas, the claws on a lobster, those little swimmerets that are at the back of a lobster's tail, those are all modified appendages. So this is what we think happened about 400 million years ago among this group of arthropods, is we had this mutation that occurred that began to develop fewer and more specialized appendages. Right? So you can see how those little changes would have affected these organisms through time, and that would have led to a bunch of differences, different types of critters. And if we look at evolution through time, we could look at follow the horse. And I put this up here because I want to remind you, of course, that evolution does not have a goal. It's not trying to produce a perfect organism. Evolution's goal is only to make natural selection anyway, is only to make the organism adapted to the life today. Natural selection just gives organisms the ability to survive well enough for today. And as time changes, or as the earth changes through time, like you see in the case of the horse, horses started off very small. They started off as browsers, all right, which means they lived in forests. And as the earth began to dry up, remember those dry areas, horses began to move out and become grazers. So they moved out into the open areas. Now, when you move out into open areas, you subject yourself to predation. Horses are usually prey, right? They're herbivores. So when horses moved from browsers to grazers, there was an evolutionary change that occurred for survival's sake. They became taller, all right? They became bigger. That's a good way to protect yourself from predation. And their legs began to change. Horses initially, of course, started off with 4 toes, just like most animals. Now, modern day horses have basically 1 toe. The other toes are there, the bones are there, but they're just small little splints, actually. The 1 toe is the middle finger toe. That's the hoof of the horse. That's what that is. That's the middle. But again, no evolutionary goal. You can see there's all kinds of horse ancestors that evolve through time, and eventually what we get is the modern day horse. And this is all driven by evolutionary changes, environmental changes. No goal, right? All right. Let's talk a little bit about evolutionary novelties. There's always this argument out there against evolution, is if evolution works by just happenstance changes, how did we get something as complex as an eye? Or among birds, how did we get feathers? Even legs. How did legs come about? And Stephen Jay Gould asks a very interesting modern day paleontologist, asks a very good rhetorical question. He says, what good is 5% of an eye? If you look at your eye today, we have a very complex eye, very complex camera eye. It's called a camera type eye. Part of that eye wouldn't work. In other words, you need all the structures together to work completely. Same thing with the feathers, and same thing with the leg. Part of a feather is not going to help you fly. Part of a leg is not going to help you walk on land. So how did these evolutionary novelties arise? How did these complex evolutionary novelties that we see around today, how did they evolve when Mother Nature only selects for those advantageous variations? And only having part of a modern eye would not be an advantage. Only having part of a modern feather would not have an advantage. Only having part of a modern leg would not have an advantage today. These evolutionary novelties we call exceptations. Exceptations are structures that evolve for one purpose, right? One original purpose, which was beneficial for the organism at the time, but became fortuitous or became a good thing to have in another context, in later time. All right, now I'm going to go into the detail of an eye and show you an example with that. But when we get into birds and we get into organisms with legs, we'll talk about this as well among feathers and legs. Now, just to give you kind of a hint, birds evolve feathers not to fly, they evolve feathers to stay warm. And then later on, feathers continue to evolve until eventually they had the ability to fly. But let's just talk about eyes in this particular example. So let's imagine a context in which eye evolution could occur. So we're going to start up at the top, and we're going to say, all right, let's start with a very simple, well, not an eye, right? Not an eye, but a series of cells, pigmented cells that were photoreceptors, so they could tell light. Now, we even see this in modern day critters. Patella, a limpet, which is a mollusk, has a eye, in quotes, has an eye like this. It's a series of photoreceptive cells that can sense light. Now, they can't sense images. They can sense shadows, basically. If you think for just a second, how would that be advantageous at the time? Well, let's imagine, I'll come back to this slide. Let's imagine you're a critter with one of these little eye spots or these pigmented cells, okay? And all you can tell is shadows. And let's say you live in the water. All right, so here's the bottom, here's the water, here's some plants, let's say, that are growing there. And you're one of these little critters swimming out here with your eye spot. Now, you're prey, and you want to make sure that you hide from predators, which would have been important, obviously. Having an eye spot wouldn't be able to for you to see the predator, but what it would be able to do was tell if a predator is passing overhead, right? As that predator is passing overhead, it would cast a shadow. Now, a shadow is not a distinct image necessarily, but it does give an indication that light has changed. If you had those pigmented photoreceptive cells, you might be able to sense that shadow, and evolution would have driven those organisms that were able to make the connection between that shadow and a predator, and maybe then you duck into the weeds so that you hide. Now, maybe later on, other species develop the ability to have these same pigmented cells, but maybe bulging out from the side of their bodies. So now they would have vision not just above them, but in more than one direction. It's still not an eye like ours. Still can't use that eye to tell one image from another, but you're sensing shadows. So you're sensing light, which is what your eye does, right? Your eye senses light. Now, what would happen if we took that eye and we turned it into an eye cup? Okay, so now we have pigmented cells in a cup, like we see, for example, in modern day mollusks, the Pleurotomeria. What pigmented cells in a cup would have done would have been very similar to the eyes bulging out from the side of the head. This one would only be able to sense light from above, whereas here you can sense light from many directions. All right? That would have been advantageous, wouldn't it? Now you don't have to worry about, now are you only limited to knowing when a predator moves over top of you. You might be able to see when a predator moves off to the side, and maybe then you duck into the weeds and hide. Okay? Now we're going to continue on with that idea. What if that cup continued to form, continued to narrow, like we see in something called a pinhole eye? Pinhole eyes are found in modern day organisms of Nautilus. Nautilus is another mollusk, and they have a pinhole eye, and they develop a fluid-filled cavity. And what this would have done, it would have increased the what we call the visual acuity. So now they may not be able to see images perfect, but they see a little more than a shadow. And the eye cup getting narrower would have been very similar to how I do it. If I wake up in the middle of the night and I look across and try to see what time it says on the clock that's on my little DVR box that sits by my TV, I can't see. I have bad vision. But if I squint my eyes, I can make out the time. And if you wear glasses or anything like that, you know what I mean? Even if you don't, if it's bright outside, you squint your eyes, you can see a little better. The squinting is narrowing down the light. You're focusing the light on your retina. And that's what a pinhole eye would have done. So now we see a change from just a cup of photoreceptive cells to a cup with a narrow opening, okay, and this fluid-filled cavity. And in that fluid-filled cavity, if we move a little bit further to part D, a lens would have developed. And we see that now in Murex, a modern modern. It's a contemporary marine snail that exists today. So now we're getting something that can focus. And then we can jump to the eye that we see today, like we see in squid, loligo, right? In squid or sharks have an eye like this, right? So here we go. This is the lamprey. They've got that optic, they have that cup, right? And then we get into the sharks, and we have a lens, and you have an iris, and you have muscles that can move the lens, and you can focus the lens, and then you get yourself your camera type eye. All right, so you see, when Stephen Jay Gould was asking that rhetorical question, what good is 5% of an eye? It's no good at all. 5% of an eye, if we're relating it to the eyes we have today, is no good. And the problem with that in evolutionary history would be that along the way, those incremental advances in making an eye would have had to convey an advantage, or they wouldn't have been selected for. That's just basic natural selection, right? But as I've demonstrated to you, each of these, the pigmented cells, the photoreceptors, the eye cup, the pinhole eye, the primitive lens, all of those would have conveyed an advantage. If you can tell a shadow, if you can tell which direction it's coming from, if you can increase the acuity, as the pinhole eye would have done, and the one with the primitive lens, and then, of course, with the complex camera eye, you would have obviously gained an advantage. You can see your environment. You can see where predators are coming from. You can see where prey are if now you're a predator. Right? So that's what we mean by an acceptation. It was something that was fortuitous at the time, and little tiny changes continued to make it more and more fortuitous in each species until we finally get to the camera type eye. Same thing with feathers and same thing with legs, which we'll talk about when we get to it. Remember, the important thing about all these, though, is the idea that each one must convey an advantage to the organism that possesses it. All right? I think that's it for this chapter. Yeah. All right. Okay, so that's it for chapter 26. That's where the exam material will be over. I do want to get into chapter 27 in the 10 minutes I have left. Just get started on it. Get that loaded. Oops, wrong one. Hold on a second here, folks. There we go. Chapter 27. The prokaryotes, bacteria and archaea. These things are found everywhere. You have 10 times as many prokaryotic cells in and around your body than you actually have cells, your own cells in your body. We say that prokaryotes are ubiquitous. Now, I will try to spell this. Ubiquitous. That's not quite right, but ubiquitous means everywhere. This actually is an electron microscope of the head of a straight pin, and all those little yellow things all over that straight pin are bacterial cells, prokaryotic cells. Tons and tons of them. Prokaryotes also, especially among some species, definitely within the domain archaea, have the ability to survive in some very, very harsh climates. I like to call them the extremophiles of the living world. For example, many prokaryotes can stand very high doses of radiation, somewhere in the vicinity of 3 million rads. A rad is a measurement of radiation. Just to give you something to compare that to, 3,000 rads to you is death. 3,000 rads of radiation would kill you. All right, and these guys can, I'm sorry, the rate, not 3,000, it's less than that actually. Well, you could do the math. They can absorb a lot more radiation than we can. 3,000 times more of radiation than we can in terms of causing fatality among humans. I'm sorry, I screwed that all up. You can read what it says there. They can live in very low pHs, very acidic, 0.03 pH, which is an acid that's strong enough to dissolve metal. These guys can live. They can live in water that is very close to boiling, above 80 degrees Celsius. They can live in extremely salty conditions. The earth or the ocean's about 3.5%. They can live in salinities up to 27. So what, about 9 times? 8 or 9 times as salty as the ocean. These guys are really, really good at living in extreme environments. And why that's important for us for the evolution of life on Earth is you got to go remember what the early Earth was like. The early Earth was that extreme. There was a lot of radiation on the early Earth. For one thing, we didn't have the ozone layer to block out the ultraviolet rays or any of the other radiation coming from the sun. There would have been lots of places where there would have been really hot water with all the volcanic activity, and these guys had the ability to survive. They would have been the first critters on the planet. And now that as we expand ourselves out into the universe and into our solar system, we suspect that other areas out there may have these types of life. They talk about life on other planets. I guarantee there's life on other planets. Now, is it intelligent, complex life like we are? Probably not. For the most part, now there might be, but for the most part, what we might expect to find would be microbial life, like you see in this picture here. Bacteria and prokaryotes don't fossilize very well for reasons that we talked about, right? They're soft-bodied organisms. But they do occasionally. So this has been suggested to be a fossil of a bacterial organism. Obviously, this has been seen under an electron microscope. It's been magnified quite a bit. And it kind of looks like the fossils of modern bacterial organisms. The interesting thing about this, though, is this particular fossil was found on a rock that we discovered in Antarctica about 20 years or 25 years ago. And geologists, for reasons that geologists can do, which I'm not a geologist, but it has something to do with rock characteristics, have told us that this rock that was found in Antarctica was at one time a meteor that came from Mars. All right, so something hit Mars and threw off a bunch of chunks of rock. A lot of that rock fell back down on the planet of Mars, but some of it got kicked up into the atmosphere so far it actually went off into space and rotated around in space for God knows how long, eventually got close enough to the Earth, it got taken in by the Earth's gravity, fell to Earth as a meteor, and here it is. So scientists were very excited when they saw this. This meteor, presumably from Mars, appears to have the fossil of a prokaryotic organism. Now, it wasn't a prokaryotic organism from Earth, it appears to be a prokaryotic organism from Mars. That really gives us an idea that maybe, at least at one time, life existed on Mars. Now, there's a lot more evidence that's been accumulated since then. To me, it just blows me away to think about my years in science, how it's gone from no conclusive proof that life exists any place else to now an awful lot of evidence to suggest that it does. Now, to be fair, geologists, other scientists have argued that geological processes, in other words, not living things, could have formed this particular structure that you see. So, it's the debate. Is this a fossil remain of some prokaryotic organism? Could be, certainly looks like it. Could it be something that was formed through geological method? Could be that as well. Remember in science, the idea of a hypothesis, you never really prove anything. You support or you refute. So there's evidence that supports that this was a fossilized prokaryote from Mars, and there's evidence that supports or refutes that. It supports that it might have been something geological. Nonetheless, these things are everywhere, not only on Earth, but maybe throughout the universe. One thing about prokaryotes, though, most of them are unicellular. Not all of them. Some species do gather together. Sometimes they get together temporarily. These is a bunch of prokaryotes that are growing on a petri dish. That's that dish that's got that gel, that agar gel that they can grow bacteria on. But most of them are unicellular. But the interesting I want you to understand here is that even in these early organisms, earliest organisms, we do see this idea of bonding together, right? Sometimes prokaryotes, even though they're unicellular, come together to form colonies, and even at times when they form colonies, they actually divide up labor amongst those cells. And the reason why I'm talking about that is if you think about it, that's what multicellular creatures do. Now, prokaryotes are not multicellular, folks. Even when they form these colonies, they're not multicellular. Multicellular has a very specific definition, all right? We, of course, are multicellular. We have tissues, and tissues are collections of cells. Prokaryotes can't form tissues. But even back then, even amongst these critters, not back then, I'm sorry, but even amongst these critters, we see in some cases this evolutionary trend, right, to live together cooperatively. So we already start to see the what you might call the precursors, or you know the what am I trying to say? I'll just stick with that. Precursors to multicellularity in tissues among these critters. All right, so the unicellular shape, they really come in three basic morphologies. They're either round, cocci, they're rod-shaped, bacilli, or they have this spiral-shaped, right, a helical or spiral-shaped. Cocci bacteria can be broken down to also into whether they are two of them together, two circles together, is called diplococci. A chain of cocci is called streptococci. And then those that grow like a grape cluster is called staphylococci. Staph aureus, if you, medical field is always dealing with Staph aureus infection. Staphylococcus aureus is a prokaryotic organism that when you look under a microscope, the round cells form cluster grapes. I don't draw clusters, I don't draw grapes very well, but there you go. All right, so you got round, rod, and spiral shaped. They're all usually pretty small, anywhere between 1 to 5 microns. The typical eukaryotic cell or the typical plant and animal cell, like you looked at under the microscope in Bio 1, is anywhere between 10 to 100 microns. So prokaryotic cells are significantly smaller. Remember, cells like to stay small because it increases their surface area to volume ratio. There are some big ones, though. This is a sulfur bacteria. There are certain bacteria that actually can use sulfur as a form of energy. Thiomargarita, makes me think of something else. Thiomargarita nambiensis is about 750 microns. There's a picture of it next to a bee to give you sort of an idea. Pretty good size prokaryote. For the most part, they stay pretty small. They have a cell wall, not the same kind of cell wall we saw in plant cells, not made of cellulose, but they do, nearly all of them possess this cell wall, and they also have, in many cases, modifications to their cell structure. And the cell wall does a lot of the same things that it did in plant cells. It maintains the shape. Physical protection, the cell wall can oftentimes help the cell hide from immune cells in an organism. Same thing that happens in plant cells. It can prevent the cell from lysing in hypotonic environments. Remember, plant cells have a cell wall. One of the reasons is because they like to absorb as much water as they can, because plants can't go get water. You know, so if they absorb too much water, they would lyse in a hypotonic environment. But the cell wall, remember, animal cells will, don't lyse. Plant cells have the cell wall. I always tell my Bio 1 students, like trying to blow a balloon up in a box. If you have a cell wall, you know, that balloon can never push, you know, the central vacuole filling it with water will never cause the cell to rupture because of that cell wall. It's obviously not the case in animal cells. Prokaryotes have that as well. But there's something interesting about the cell wall. Among many forms of bacteria, there are bacteria that we call gram-positive and gram-negative. And I'll talk later about where the gram-positive and the gram-negative comes from. But let's look at the cell wall structure. Here's the plasma membrane. Okay, that plasma membrane is the same plasma membrane you learned about in Bio 1. But among prokaryotes, there's also another layer that's called a peptidoglycan layer. In gram-positive bacteria, we have a plasma membrane and a thick peptidoglycan layer on the outside. In gram-negative bacteria, we have 2 plasma membranes, and in between, sandwiched in between, is a thin peptidoglycan layer, right? Underneath the microscope, their names gram-positive and gram-negative come from the way they're stained. You can tell a gram-positive bacteria because when it's stained properly, it'll be purple. Gram-negative ones will be that light red color. Now, one more thing, and I'll leave you for the day. On the outer membrane of gram-negative bacteria, and this is why many gram-negative bacteria tend to be the ones that cause disease. Many gram-negative bacteria have lipopolysaccharides, little green things that are sticking up. Lipopolysaccharides, fats and sugars attached together. And many times in species, lipopolysaccharides produce endotoxins. Toxins, those are the things that make you sick. For example, with the tetanus bacteria, what causes the muscle seizures, the lockjaw part of tetanus, is the endotoxins that are produced by the lipopolysaccharides. All right, well, I think I'm out of time for the day. We'll pick up next time, and I'll discuss how we stain these to get them gram-positive and gram-negative, and then we'll discuss the rest of these really bizarre, interesting critters, all right? So let me stop the recording.